Okay, hello to all of you. Let's discuss the second part of chapter number 6. So let me go into the slideshow mode. So in this part, we are going to discuss about cytoskeleton, and also we are going to talk about what is outside the cell, and we are going to talk about some of the junctions which are between the cells. So first of all, what is cytoskeleton? So cytoskeleton, if you can break this word into two parts, cyto means cell, skeleton means skeleton. So cytoskeleton is the skeleton of the cell. So just the way we have our skeleton, we have the skeleton of our body, like we have bones, we have muscles, ligaments. So all these things, what do the muscles and bones do? They give us a shape as well as they help in the movement. So similarly, at a cellular level, at the cells level, there is a skeleton, and that's cytoskeleton is made of some protein filaments. So we are not going to go into a lot of details of the protein filaments which make the cytoskeleton. So basically you guys need to know the names of these three protein filaments. You need to know some of the important characteristics and their function. That's all for you guys. So basically they are microtubules, microfilaments and intermediate filaments. So this is basically showing a picture in which fluorescent staining is done, and it is showing different cytoskeleton filaments, protein filaments. Okay, so microtubules, they are the thickest of the three components. Microfilaments, they are the thinnest, and intermediate filaments, as the name itself says, their diameter is in between. Okay, now this is an important slide because this actually has all the information. So first of all, we have microtubules. So they are like hollow tubes. So you can see they are like this. Okay, so they are like, these are hollow tubes, and they are made of a protein called tubulin, and tubulin is a dimer consisting of alpha tubulin and beta tubulin. Okay, so they are dimers. They are made of alpha tubulin and beta tubulin. So alpha beta, this is a tubulin dimer. Okay, so some of the important functions of the microtubules. So these microtubules are involved in the chromosomal movements during mitosis. So we will be, when we will talk about cell division, we will discuss it in more detail. So these microtubules, they help in the separation of chromosomes during the cell division. The mitotic spindle, which is made and which helps in the separation of the sister chromatids as well as the chromosomes, it takes place with the help of the microtubules. Also cilia and flagellum, they are the kind of appendages. Cilia and flagellum, they help in the movement of the cells. They are involved in cell motility. They are also made of microtubules. Then microfilaments, they are the thinnest protein filaments which make the cytoskeleton. They are basically two intertwined strands of actin. So the protein involved in microfilaments is actin, and they are, you can see, like these are two intertwined strands of actin. Okay, so they are involved in muscle contraction. They are also involved in cytoplasmic streaming. So I don't know if you have seen in the lab in maybe biology 1101 or in any other class, like if you take a leaf and you put it under the microscope and you look under the microscope, if you see the cells inside the cells, in the leaf cells, you will see lot of chloroplasts. And you will see that you can see, I don't know if you've seen that, the chloroplasts are moving actually, and this phenomena is called cytoplasmic streaming, and this takes place with the help of the microfilaments. They are also involved in cell motility, for example amoeboid movements, like some of the lower organisms, they can move and they make amoeboid movements. That's also because of the microfilaments. Then the last one is intermediate filaments. So their size is in between, the diameter is in between, and they are basically fibrous proteins coiled into cables. So they are like this. So they are fibrous proteins which are coiled into cables. They are basically involved in the maintenance of cell shape, anchorage of nucleus and certain other organelles, and also formation of nuclear lamina. So the nuclear envelope is basically made of nuclear lamina, and the nuclear lamina has intermediate filaments in it. So that's all basically you need to know about these three cytoskeleton protein filaments. Now we're going to talk about the extracellular components, means what is outside the cell. So now plant cells and animal cells are different. In the plant cells, outside the plasma membrane, there is a cell wall. Okay, and in animal cells, they do not have the cell wall. So outside the cell, they have extracellular matrix. We call it ECM. And ECM is made of many glycoproteins. Okay, so we're going to talk about those. Now, first of all, let's talk about the plant cells, the cell wall in the plant cells. Now, the cell wall is not only present in plant cells, it's also present in the bacteria. I showed you in the last lecture, in the last video, that most of the bacterial cells, they have a cell wall. Even fungus, they have cell wall, and plants, they have a cell wall. So the cell walls protect the plant cells, maintain its shape, and prevent excessive uptake of water. In different organisms, the cell walls are made of different chemicals. Like in the bacteria, it's made of peptidoglycan. In fungus, it's made of chitin, and in plants, it's made of cellulose. So let's see the structure of the cell wall of the plants. So it has multiple layers. It has a primary cell wall, which is relatively thin and flexible. Then middle lamella, which is thin layer between primary walls of the adjacent cells, and the secondary wall, which is added between plasma membrane and the primary cell wall. So you can see here, this is the primary cell wall, and primary cell wall is like thin. This is the secondary cell wall, which is thick. So this is one cell. So this is primary cell wall, this is secondary cell wall, this is another cell. So this is primary cell wall, this is the secondary cell wall, and in between these two cells, in between, this is called middle lamella. So now let's talk about animal cells. So animal cells lack cell walls, but they have extracellular matrix, and this extracellular matrix is made of glycoproteins such as collagen, proteoglycans, and fibronectin. And also the proteins which are present in the extracellular matrix, they bind to the receptor proteins which are present in the plasma membrane, and those receptor proteins are called integrins. Now you can see here, this is an animal cell, this is the plasma membrane, this is the phospholipid bilayer, and these are various proteins which are embedded in the phospholipid bilayer. And one of the protein is integrin. So integrin is a receptor protein which is embedded in the plasma membrane of the cells. It's a receptor protein. And outside the plasma membrane, this all region is called extracellular, it's called an extracellular space basically. And in this space are secreted a lot of glycoproteins like collagen, fibronectin, and these ECM, it makes, they interact, all these proteins in the ECM, they interact with the integrins, which are the receptor proteins embedded in the plasma membrane. Oops, sorry. Oops, sorry. Okay, going back, we were here. So ECM proteins bind to receptor proteins in the plasma membrane called integrins. So these are all ECM proteins, collagen, fibronectin, they all interact with proteoglycans, they interact with these integrin proteins. These are the receptors. Okay, now we are going to talk about the cell junction. So how these cells, they are connected with each other. So in the plant cells, there are some cell junctions called plasmodesmata, So these plasmodesmata are basically channels through which the two plant cells, they can exchange various material, like they can exchange water, they can exchange proteins, RNA between them. So these are the channels through which the two plant cells, they can exchange material between each other. In the case of animal cells, we are going to talk about, there are three different kinds of cell junctions we are going to talk about in the animal cells. They include tight junctions. So the tight junctions are those junctions between the two cells which are so tight that nothing can go across through these tight junctions. Then there are desmosomes. Desmosomes, they are basically anchoring junctions, and they basically fasten the cells together into strong sheets. Basically, these are like anchoring junctions. They anchor the two cells together. And the last one is gap junctions. So gap junctions are just like plasmodesmata in the plant cells. So the equivalent term of plasmodesmata in plant cells is gap junctions in the animal cells, because in the animal cells, through gap junctions, they are like channels through which these cells, they can exchange material between them. So here you can see these are the animal cells. So this is one cell, behind is a second cell, and here is a tight junction. So means tight junction is that part between the two cells where these cells are anchored so tight that they do not exchange anything between them. Here they are very, very tightly bound to each other. Then the desmosomes, these are called anchoring junctions. Here the two cells, they are anchored to each other. Gap junctions, you can see these are channels here through which the two cells, they are exchanging material between them. So that's all about this chapter. This finishes this chapter. So if you have any question, please let me know.